Hi, I'm Jim Sandstrom. I'd like to talk to you about SRP technology. What is it? What makes it special? But first I'll start with a question. Do you recognize this small item? I've drawn it here in a larger version to make it a little more clear. You've seen this 1,000 times in your life, but I bet you don't remember it and don't recognize it. On the other hand, you recognize these immediately. You know a football, a basketball, and a volleyball when you see it. Now, what's the purpose of this question? I'd like to ask you, what do these three balls have in common? Well, it's the air valve. Each of these balls, although they're different shape and color, they each have an air valve. And the air valve is extremely similar between all three balls. What I'm doing today is using this analogy of the immune system and how the immune system recognizes bacteria. "20 years ago, scientists at Vaxxinova, US, formerly called Epitopics, looked for a commonality between bacteria. And what they chose as a common factor between all bacteria that's quite similar is the SRP protein. SRP protein in this illustration is the air valve. I'm going to show you how that applies to microbiology. When the immune system looks at a gram-negative bacteria, the most distinguishing characteristic that it's going to recognize and remember is the lipopolysaccharide. That's a long-chain sugar, or polysaccharide, attached to a lipid molecule called lipid A in the cell membrane. We also call this LPS. Another name for lipopolysaccharide is endotoxin. That's right, the same chemical that we think is sometimes harmful to animals in a vaccine reaction is the active ingredient of conventional whole cell bacterins. The immune system recognizes the endotoxin and makes an immune response to it. Another name for the same structure is the O antigen. This is why we have E. coli O157, E. coli O78, or O1 or O2. There's a couple hundred types of E. coli, each one with a distinctly different polysaccharide pattern that the immune system can recognize and remember. There's one more synonym for the same molecule. Serotype. The reason we call bacteria serotype, such as Salmonella typhimurium, that's the serotype. Salmonella Dublin is a serotype. We have 12 or 15 known serotypes of Pasteurella, each one distinctly different from the other based on their polysaccharide, and that's what the immune system remembers. But remember now, we are trying to come up with a commonality between these bacteria. And so now I'll show you what it is that SRP does that is unique for the bacteria and makes it a great vaccine. When a gram-negative bacteria such as E. coli or Salmonella enters a host animal and begins to cause disease, it needs to acquire iron from the host. Iron is essential for bacterial growth and survival, just the same way that oxygen is essential to the host animal. To acquire this iron, it needs to take it away from the host proteins such as hemoglobin and lactoferrin. It does it by secreting siderophores into the environment of the bacteria. The siderophores find iron bound to the host proteins, and they take it away from the host protein and they make it available to the bacteria. The siderophores will match up with a siderophore receptor protein and bring the iron into the cell. That's a very important function for this SRP molecule, but the best part of all is that it's the commonality between bacteria, and it's something that the immune system can remember that is independent of what bacteria it comes from. When we make SRP vaccine, we go through a special step that conventional bacterial vaccines don't need to do. In making SRP vaccine, we break apart the whole bacteria and we do several steps of manufacturing to purify the SRP and to get rid of almost all of the endotoxin from the vaccine itself. That's going to make the vaccine less reactive than a vaccine that has a lot of endotoxin in it. It's also going to give the immune system a chance to really focus on what's important, and that is the SRP that's common between various types of bacteria. Now let's tie it all together. We learned a few things about SRP when we started making vaccines. The first thing is that bacteria like Salmonella all have the same SRP types in common. In other words, whether we take Salmonella typhimurium from a poultry farm or Salmonella Dublin from a dairy farm, the SRPs are exactly the same. Salmonella always has the same 3 SRPs, identical. That makes it really easy for the immune system comparing one Salmonella serotype to another. Another thing we learned is the air valve principle, the commonality, that SRP-B, for example, is identical whether it comes from a Salmonella or a Klebsiella or an E. coli. And that means if you have the right SRPs in the mix, it really doesn't matter which bacteria they came from. The final thing I'd like to tell you is best illustrated by telling you about a study done at Iowa State University in the Iowa State dairy herd. We made a Klebsiella vaccine to protect against Klebsiella mastitis in the dairy cows. It was very effective against Klebsiella, but the surprise in the results is that it was also quite effective against E. coli mastitis at the same time. And you can see why. It's because of the common SRPs between Klebsiella and E. coli. And this is an SRP principle that's been very important and very beneficial to many animal species. So in summary, I'd like to say the reason we make SRP vaccine instead of conventional whole cell Bactrians is because the immune response is broader than whole cell bacteria, because the SRPs can be found across a wider range of bacteria than making vaccines that are dependent on LPS. Reason number 2 is that by getting rid of the endotoxin as we're manufacturing the vaccine and just keeping the proteins, we have a vaccine that is less likely to cause a granuloma at the vaccine injection site and also less likely to suppress the activity of the animals and their appetite in the days following vaccination. If you'd like to know more about SRP technology vaccines, please talk to your Vaxxinova representative or visit our website at vaxxinova.us.com. Thank you.